you for joining today. We are the DEI working group of BINA. And so every month we are organizing different events. This is February right now, so it's Black History Month. And so we are very, very happy to welcome, to host Katiannah Moise. She did her bachelor's degree at Pomona College, then a postbac at the University of Chicago. Then she completed a management course at this prestigious Kellogg School of Management at Northwestern University. And this was while she was doing her PhD also at Northwestern University. She does really cool work on vasculature. So I'm really excited to see that. She recently defended her PhD thesis. And I think a lot of what she will tell us today is precisely about that. So I'm very excited to hear her talk.
Thank you, everybody. Okay, so I will share my screen. All right. So I'd like to thank everybody for joining me, the community of BINA. I also like to thank Mariana for inviting me and basically introducing me to the BINA community. And I'll talk about my thesis dissertation that I did at Northwestern University. So first and foremost, all cells are exposed to physical and mechanical forces, and these forces actually inform how cells adapt and behave to their microenvironment. And these different cellular behaviors occur through a process called mechanotransduction, which cells essentially read mechanical cues from their environment that then gets transduced to biochemical signals and then eventually is used to regulate force signaling pathways. And endothelial cells are no stranger to being exposed to different forces. And as a vascular biologist, I actually study the ways in which endothelial cells are exposed to shear stress and how that affects their cellular behavior. And one of the most very important blood vessels in the mammalian animal is actually the aorta, which extends out from the heart. And within all blood vessels, the innermost layer of the vessel is made up of endothelial cells. And in our aorta specifically, it's very interesting because the architecture, which is that curved candy cane look, is exposed to different types of shear stress or mechanical forces. And they're described here on the right, where certain regions are exposed to undisturbed continuous laminar shear stress or disturbed multi-directional forces. And then you can actually parcel them out based on their dynamics, whether the forces are steady state, pulsatile, or oscillatory. What's more important to know is that endothelial cells have this very sophisticated mechanism in being able to recognize different types of forces. And so here is a video of endothelial cells basically being exposed to continuous laminar shear stress. And you can really see that they are dynamically changing their cell shape and their direction in order to properly respond to this force. And so when we look at the aorta and you actually look at the architecture, as I described before, that in regions of the vessel where it's curved or bifurcated, endothelial cells are actually exposed to multidirectional disturbed forces, while endothelial cells that are in the descending aorta, or basically where the aorta is straight, endothelial cells are exposed to laminar uniform flow. And because of that, they actually acquire a phenotype where they basically are very elongated, and they also are always parallel or following the direction of flow. The way that they respond to shear stress is actually linked to vascular disease. So as I said before, when endothelial cells are exposed to disturbed flow, which is usually happening at curved or bifurcated regions of vessels, these endothelial cells actually acquire a rounded or polygonal phenotype. And in the context of vascular disease, specifically atherosclerosis, it was found that these endothelial cells actually take on a pro-inflammatory profile and then are susceptible to the development of atherosclerotic plaques. And because of that understanding, there are a whole host of vascular diseases where it's been shown that these endothelial cells have these dysfunction that is characterized by abnormal cell shape. And then that leads to furthering dysfunction related to mechanoresponse. And these other diseases include hereditary hemorrhagic telangiectasia, which basically is a genetic disorder where the endothelial cells are larger than they're supposed to be and they can't align, and therefore the formation of the vessels create these large bulbous regions that are very susceptible to hemorrhaging, and that's why you have a lot of bleeding. And then also in the case of pulmonary arterial hypertension, endothelial cells don't function as well because they also show maladaptive responses to flow. And so the ability for endothelial cells to properly respond to flow is mediated by cytoskeletal remodeling. And so the cytoskeleton is essentially composed of three main filaments, which are microtubules, intermediate filaments, and actin, which interact with one another and are responsible for cell shape-based functions related to migration, adhesion, stiffness, and intracellular trafficking. And here I study microtubules because a lot less information is known about the role of microtubules and its regulation and understanding how they're necessary for how endothelial cells are respond to shear stress. And so microtubules are rigid rods made up of 13 filaments stacked up into a cylindrical shape, and their stability and length is basically regulated by cycles of polymerization or depolymerization under the umbrella of microtubule dynamic instability. And this ability to regulate the filaments are basically regulated by many different proteins. And one of the ways that I'm studying this regulation is through the concept of these post-translational modifications, which are essentially different residues that are reversible that are added to the C-terminal end of tubulin tails. And these modifications are really important because they're known to regulate stability of microtubules. They help direct and dock proteins and microtubule-associated proteins to specific parts of the microtubules. And essentially, they're also really important in those cellular behavior processes related to division, migration, and intracellular trafficking. So essentially, what is known is that these post-translational modifications are what confer diverse properties to microtubule functions. And it's really important to note that it's been shown that when the distribution or the expression levels of these modifications are abnormal or altered, they have been implicated in disease and dysfunction. And most of the research has been around trying to understand what occurs with intracellular trafficking and responses to forces, but it's really well known that when you alter these modifications, they lead to diseases such as in cilia disorders, in neurodegenerative diseases, and also in cardiomyopathies. And so in keeping in line and trying to understand the role of microtubule dynamics in endothelial cells, I'm trying to understand what are the molecular mechanisms being driven in regulating endothelial cells to allow these cells to basically respond to flow. And then furthermore, trying to understand what is the role of microtubule dynamics and better understanding the role of cell shape in dysfunctions related to vascular diseases, as I talked about in atherosclerosis and hereditary hemorrhagic telangiectasia. And so the very first thing I wanted to do is try to map out the parameters and studying functional endothelial cell responses to flow. As I said earlier, that when endothelial cells are under laminar continuous shear stress, they elongate and align in the direction of flow. So we're going to be looking at elongation factor, was basically cell shape, looking at major axis and minor axis of the cell. And then we're going to look at alignment, which is essentially just the orientation of a group of cells. And from there, we can actually target different aspects of microtubule dynamics to understand how disturbing them affects endothelial cell responses to flow. And here I'll go through looking at the effects of microtubule targeting agents, which disrupt the cycles of polymerization and depolymerization of microtubule filaments, looking at disturbing the distribution and localization of post-translational modifications, and then also looking at the role that the enzymes responsible for regulating these post-translational modifications, how do they affect microtubules and thus endothelial cell responses to flow. So in this whole thesis project, we used a cellular segmentation algorithm called CellPose. There are so many different types that exist, but essentially what this tool does, like Fiji and Cell Profile, is basically is used as a tool to gather large data sets from immunofluorescence images. And so here, working with the Goyal lab, what they were able to do is to take my immunofluorescence images of endothelial cells stained for VE coherin which is a proxy for cell shape, and actually use that to be able to create these predictive masks that would give you these major and minor axis data, which would then give us values that we can use to understand the nuance of disrupting certain aspects of microtubule dynamics and its effect on cell shape and alignment. And so the very first experiment we did was try to understand the relationship between endothelial cell responses to flow and microtubule reorganization under shear stress. And so here you can see is a time course of endothelial cells exposed to laminar shear stress for 48 hours. And what you can really see clearly is that under still conditions or static, endothelial cells are rounded and polygonal, but over time, the longer they are exposed to flow, they begin to dynamically change their cell shape and become more elongated and aligned in the direction of flow. When we stain for microtubules using this live red tubulin dye, we can see that at still conditions, the microtubules are more astral, they're shorter, but over time we see that there's a bundling and lengthening of these microtubules within these elongated endothelial cells. And this video here really demonstrates the relationship that we see between these changing endothelial cells and their cell shape to microtubules and their reorganization. So here you can see in green the microtubules, and then the endothelial cells cell shape aligned in red under shear stress. And so the very first experiments actually look at the relationship of targeting microtubule dynamic instability and its effect on endothelial cells. And so here we used microtubule targeting agents, and essentially these agents block one aspect of microtubule dynamic instability. So here, nicotazole and colchicine basically block polymerization, so it prevents the addition of tubulin to microtubules, while Taxol prevents the removal of tubulin from microtubules. And what's been known is that these targeting agents were first found as a way to target cancer cells, because it's been known that when you pre-treat the cells with these microtubule targeting agents at high concentrations, it leads to mitotic arrest and cell death. But what's interesting is that at sublethal concentrations, these targeting agents can actually be a really great tool to understand how just disrupting microtubules without completely obliterating the microtubule network can help us understand its important role in endothelial cell function. And so in order to actually study that relationship between microtubule dynamics without killing the cells, we actually have to confirm that we're able to treat these cells, stop or impair microtubule dynamics without affecting endothelial cells. And so what I did here is that I actually transfected endothelial cells with EB3, which is essentially stands for end-binding protein 3, which is a marker for polymerization of microtubules. And what we can see here on the bottom is that I did these live cell images to see what its effect on disrupting microtubule dynamics by either affecting the morphology, the kinetics of this EB3 marker. So here is a really nice video of just regular control cells transfected with EB3. You can see these beautiful comets that look like fireworks, and you can really see very quickly that these comets basically run all the way from the microtubule organizing center of the endothelial cell all the way out to the periphery of the cells. When we treat with Taxol, which is one of the microtubule targeting agents, we can really see there's a difference. The size of the markers is different. They're much smaller. But not only that, we see that there begins to be shuttling or difference in the kinetics of this EB3 marker, indicating that there's impairment in microtubule dynamics. And we see this also with the treatment with nicotazole, and then also when we treat with colchicine. In this case, colchicine is actually really effective in obliterating the marker for EB3, indicating that there's severe impairment of microtubule dynamics related to polymerization, which makes sense because it blocks polymerization. So from there, we actually treated these mono layer of endothelial cells and want to see whether these microtubule dynamics are required for proper endothelial cell elongation and alignment. So at static conditions, comparing the microtubule targeting agents to control, we can see that generally endothelial cells will have the same cell shape and orientation, and because there is no shear stress. But looking at flow, we can really see a difference. So endothelial cells underflow properly elongate and align, which we see here, But when we treat them with Taxol, colchicine, or nicotazole, we can see that there is a reduction in cell shape and also misalignment in response to flow. And when we look at the data that was composed and calculated by CellPose, we can really see this effect. Under static conditions, endothelial cells, regardless of treatment, are randomly oriented in terms of their orientation around the 360-degree axis. Well, under flow, we can see that endothelial cells show that directionality, that paralleled alignment with laminar shear stress, and then this gets completely disrupted when you treat with Taxol, colchicine, or nicotazole. When we look at cell shape, as defined by elongation factor, we can confirm once again that generally under still conditions, all this endothelial cells are pretty much not elongated, they're polygonal. But under shear stress, we see that this flow-induced elongation is completely abrogated when we treat with Taxol, colchicine, and nicotazole, which shows to us that microtubule dynamics are really important for endothelial cell elongation alignment. And specifically, we show that just blocking one side of that dynamic polymerization, depolymerization cycling causes the same effect. We lose the ability to elongate and align, showing us that we do need that constant cycling of microtubule dynamics to allow for endothelial cells to properly respond to shear stress. So then the next thing I want to do is actually look more into those post-translational modifications. So here, looked at 4 key modifications. I looked at detyrosination, which is removal of tyrosine residues from the C-terminal tail, tyrosination, which is the addition of tyrosine residues to C-terminal tail, polyglutamylation, which is essentially different chains of glutamic acids added to tubulin, and then the more unique one is acetylation, which is a specific acetyl group that's added to the lumen or inside facing of microtubules. And essentially the question I want to ask is, one, how does fluid shear stress alter the distribution of these modifications, and also whether these modifications are important in the process of how endothelial cells respond to shear stress. So the very first modification we looked at was detyrosination, and all the post-translational modifications are marked in red. And we can see here is under static conditions, detyrosination is basically found ubiquitously throughout the endothelial cells, but as puncta. But under flow, we see that this dynamically changes and they become more distributed and more concentrated down the midline of these elongated endothelial cells. When we look at the expression levels comparing static versus flow conditions under a duration of time of 48 hours, we can see generally that there's a dip in the expression of detyrosination, but it is not affected by how long these endothelial cells are under flow. It's basically non-significant. In looking at polyglutamylation, we can see it's very different from the first modification. Here we can see that this modification is very tightly overlined with total tubulin in green, as you can see here, with a strong concentration of modification near the perinuclear area of the endothelial cells. But under shear stress, we see there's the very significant relocalization down the midline of the endothelial cells with a very strong concentration towards the midline center where the nucleus is. But when we look at the expression levels, we notice that there is no change in the levels of this modification. There's just only dynamic reorganization of the localization of polyglutamylation. In looking at tyrosination, we can see here there's a very strong overlap with total tubulin, and this remains consistent even under flow. And this makes sense as in mammalian cells, the most ubiquitous and common form of post-translational modification on microtubules is tyrosination. And we can also see consistently that even under flow, as compared to shear stress, the levels don't really change in respect to whether or not there is fluid flow. The last modification was looking at acetylation. And here we can see something very, very special in that under static conditions, you can see that this modification is very lowly expressed and is only concentrated really near the perinuclear space of the endothelial cell, most likely near the microtubule organizing center. But when endothelial cells are exposed to shear stress, we see that there's an increase in the expression of the acetylation, but also we see that it's basically found down the midline and all around the elongated endothelial cell. When we look at different endothelial cells under 48 hours, we can see that the acetylation marks are very heterogeneous and different. So in some cells, acetylation is very little. In other, you find it ahead of and behind the nucleus. And in other cases, you see a lot of acetylation marks. And when we look at the Western blots comparing static to flow, we can really see that yes, at still conditions, acetylation is very low, but once there is the onset of shear stress, we see an upregulation of acetylation, and this is consistent throughout the 48 hours of flow undergoing in these endothelial cells. In looking at the immunofluorescence time course that we did, we consistently showed that yes, under still conditions, there's very low acetylation, and then there's dynamic reorganization of these acetylation marks as these endothelial cells are elongating in response to flow, as well as an upregulation in acetylation as compared to the static conditions. The next thing I want to do is see whether this difference in acetylation due to differences in the types of shear stress that we're seeing is also replicated in the aorta architecture. So here, as I said before, that in the aorta, certain regions are exposed to disturbed versus laminar flow. So looking back at the arch versus the straight portion of the vessels, which is here in this case of the descending aorta, we can see, yes, there is a difference. So in the arch where there is disturbed flow, we see lower levels of acetylation, while in the descending aorta where there is laminar flow, we see higher levels of acetylation, indicating that there is a sensitivity in endothelial cells in respect to acetylation with different types of shear stress. So then the next step we want to do is try to understand what could be regulating these differences in shear stress that we're seeing as related to the time dependency that we're seeing. So bringing back the Western blot, looking at the levels of acetylation, we want to look at the two main enzymes that are known to regulate microtubule acetylation, which is HDAC6, which is a deacetylase, and alpha-TAT1, which is an acetylase. So looking at some transcriptomic data comparing endothelial cells that are subjected to flow, we see that in the mRNA levels show that there is basically no difference in HDAC6 and alpha-TAT1 in respect to how long they are under shear stress. So it really indicates that there might be something happening with the regulation of the proteins themselves and not their mRNA. And so in looking at HDAC6, we really do see something very unique in that under static conditions, there is higher levels of HDAC6, which is corresponding to what could be the low levels of acetylation that we're seeing in endothelial cells. And then at the onset of flow, there's a switch. So while we see here there is upregulation of acetylation, this corresponds to very low levels in HDAC6 that we're seeing here, and that this trend matches over the 48-hour period, indicating that there's an inverse relationship with microtubule acetylation and HDAC6 levels. And so you may be asking, why didn't I look at alpha-TAT1? Well, couldn't find any commercial antibodies to actually show whether there's an effect also in alpha-TAT1. But what we can really see is that there is a huge effect in the amount of HDAC6 in the endothelial cells at these specific time points as it relates to microtubule acetylation. So then the next thing we want to do is actually understand what happens when we alter the levels of acetylation in these microtubules. And so I use two different pharmacological inhibitors to block HDAC6 and alpha-TAT1. So tubicin blocks HDAC6 activity, which presents acetylation being removed, and then 5Z7-oxazole is actually a TAC1 inhibitor that is important for alpha-TAT1 mediated acetylation. So here what I did was actually compare a time course of acetylation and looking at cell shape and alignment of endothelial cells treated with tubicin as compared to controlled DMSO treatments. And so here we can see easily two different things, that with endothelial cells treated with tubicin, we see that there is upregulation of acetylation, and this happens very, very quickly, as soon as 8 hours, and there's much, much higher than endothelial cells, even at any of these time points that are represented below at half an hour, 8 hour, 24, and 48. But not only that, we see that there is alignment that occurs much sooner than it would traditionally be seen in endothelial cells. In endothelial cells, the majority of all cells are aligned with one another in respect to flow after 24 to 48 hours. But here we're seeing it as soon as 8 hours. But we saw that was very, very, very surprising is that endothelial cells under still conditions have very low levels of acetylation, but we don't see that in tubicin-treated conditions. These endothelial cells are hyperacetylated, but also they show increased cell length. When we look at the Western blots, we really see very well that just 30 minutes using this inhibitor for HDAC6, we get upregulation of acetylation about 500 times higher than even the control flow endothelial cells, and that this increase in acetylation continues throughout the 48-hour time period. In looking at the other side, what happens when you inhibit alpha-TAC1 using that TAC1 inhibitor? We can see here very quickly that even after 48 hours of flow, all these endothelial cells seem very impaired or blind to the effect of flow. They are rounded and polygonal, while in the control cells, as I said before, they usually are elongated and aligned. But not only that, we seem to lose that dynamic redistribution of those acetylation marks within those endothelial cells. When we look at the protein levels, comparing control endothelial cells with those TAC1 inhibited endothelial cells, we can really see that flow-induced acetylation is completely abrogated when you treat with that TAC1 inhibitor. When we look at alignment and elongation factor, we can see that the data really shows that when there is no flow, all the endothelial cells are randomly oriented. But when you are under flow, we see that the inhibition of HDAC6 does not affect the ability of these endothelial cells to align in the direction of flow, but this is affected when you block alpha-TAC1 with that TAC1 inhibitor. And then looking at cell shape, you can really see that without shear stress, tubicin-treated endothelial cells are much more elongated than their control counterparts, while the TAC1 inhibited endothelial cells basically don't respond and remain polygonal. And then under shear stress, we see that there is that flow-induced elongation. This is much higher when you treat with tubicin, and this is completely blocked when you treat with that TAC1 inhibitor. The next thing I want to do is actually understand a little bit more about that specific post-translational modification and what it does in endothelial cells. So people who study microtubules know that acetylation is considered a marker of stability, and meaning that microtubules are more likely to have acetylation marks, are considered more resistant to mechanical or chemical breakage. But in the field, there is like a chicken or the egg scenario because people don't know if acetylation is actually the cause or the consequence of having stable microtubules. So here in this experiment, what we want to do is actually understand what happens when you alter the levels of acetylation in endothelial cells and what is its effect against microtubule targeting agents. As we've seen before, that just altering acetylation shows that just too much acetylation causes endothelial cells to quickly elongate and align more than usual, and that too little it basically completely impairs the ability of endothelial cells to respond to flow, indicating that there is a Goldilocks effect happening where you need some amount of acetylation needed for endothelial cells responding to flow. But here in this case, we're trying to understand when you alter these levels of acetylation as a proxy of trying to increase the proportion of stable microtubules, does that actually have an effect on this characteristic of being resistant to chemical or mechanical breakage, and how does it affect then downstream endothelial cell responses to flow? And so here in this first graphic, we're looking at what happens when you pre-treat with tubicin. As we've shown before, pre-treating with tubicin increases acetylation and then causes hyper elongation. And you can see this here with the top row, looking at still conditions, that the endothelial cells that are pre-treated with tubicin are elongated. But when you treat them with the microtubule targeting agents, we can really easily see that it affects cell shape and alignment. And then under flow, we can see the very same pattern that hyperelongation that we have known very well to occur is completely lost when you treat with the microtubule targeting agents. And the data reflects this as well. Under flow, we can really see that tubicin-treated endothelial cells are elongated, and then with the treating of these microtubule targeting agents, we completely lose that cell shape, and the effect is actually so drastic. It's basically blocks the it's almost as lower or even lower than endothelial cells that were just under flow. In looking at the other side, looking at the effect with TAC1, we can see under the static conditions that endothelial cells treated with TAC1 don't respond to flow very well at all. And this is consistent even when you treat with microtubule targeting agents. Under flow, we see that the endothelial cells that are untreated with drugs align very well, elongate very well, completely blocked with a TAC1 inhibitor, and the pattern is not affected even with the microtubule targeting agents. And the data also reflects this as well, specifically under flow, where we see that flow-mediated elongation, there's an increase in cell shape, completely lost with the TAC1, and then also completely blocked with the microtubule targeting agents. And when we're looking at alignment, we see once again, without flow, all the endothelial cells are randomly oriented. So there's nothing to really look at. But under flow, we can see once again that the inhibition of HDAC6 doesn't affect its ability to align as well as the controls endothelial cells, but it does when you block alpha TAC1. And then even in every case that we're looking at these different microtubule targeting agents, we lose that directionality. And so what this is saying is that when we're trying to understand the properties of acetylation, we see here that proper microtubule dynamics, that cycling, as I was talking about earlier, is the mechanism that's necessary first and foremost for allowing endothelial cells to respond to flow. But it might be that acetylation itself is a fine tuner of endothelial cell responses to flow, as the previous experiments has said that just altering the distribution of the levels of acetylation affects how soon and how well endothelial cells elongate and align. And so in this case, in trying to answer the chicken or the egg, in the context of endothelial cells, it might be that acetylation in endothelial cells is the egg, in that you need to have proper stable microtubules first, then you get acetylation, and then you're able to have proper endothelial cell responses to flow. So then in the last experiment, wanted to further look at looking at the molecular regulators directly by blocking or genetically inactivating HDAC6 and alpha TAC1 and trying to look at its effect on endothelial responses to flow. And so here we use a CRISPR-Cas9 technology, and we're able to create these knockout cell lines, and then we put them under flow and try to look at the relationship between microtubule acetylation and its effect on cell shape and orientation. And so here on the top row, we can see very well, as we've shown before, that there's a basal level of acetylation in endothelial cells under still conditions, and then under flow, there's an increase in acetylation, and that corresponds to elongation and alignment. When we knock down alpha TAC1, we see a complete loss in acetylation, and then even under flow, where we would see flow-induced increase in acetylation, we don't see that at all. It's completely lost as well in these alpha TAC1 knockout cells. With the loss of HDAC6, we see that hyperacetylation, but also we see that there is an increase in cell length, and that increase in cell length is maintained under flow as they are more elongated and aligned than their control counterparts. What's really interesting to see is that with the loss of alpha TAC knockouts cells, we can consistently see that there is a lack of cell shape elongation even under flow using these immunofluorescence, performing immunofluorescence staining. In looking at the knockout validation, we're confirming that those knockouts that we created are true. The loss of alpha TAC1 really causes complete loss in acetylation in comparison to the control endothelial cells without affecting the levels of total tubulin. And then with the HDAC6 knockout cells, just knocking down knocking down HDAC6 causes such a strong increase in acetylation, it completely obliterates all the basal levels of acetylation that could be seen in a control endothelial cell, and then this corresponds to a complete loss in HDAC6. When we look at the alignment data, we can see once again that that loss of alpha TAC1 causes misalignment, but the effect is not seen when you lose HDAC6 here. And that with cell shape, once again, that the loss of alpha TAC1 under still conditions shows maybe a slightly more elongated phenotype in comparison to control, but for the most part, they're very rounded and polygonal, but the loss of HDAC6 really drives an increase in cell length as compared to the other two treatments, the other two knockout cell lines and controls. And then under flow, we see even more exacerbated phenotype that the knockdown of alpha-TAT1 really affects cell shape, and that this is the opposite that's seen when you lose HDAC6. And so in conclusion, I told you a lot of things today, but here we really should see a relationship between microtubule acetylation and endothelial cell responses to flow in the context of responding to shear stress. I've shown to you that microtubule dynamics are really important for endothelial cell responses to shear stress, and that there is a proper amount or a Goldilocks effect that's needed in allowing endothelial cells to not only have the right amount of microtubule acetylation, but also to properly and have timed proper responses to flow. And that when you knock down HDAC6 and alpha-TAT1, it affects their ability to elongate and align, and that acetylation is actually a fine tuner of endothelial cell responses to flow. And so I'd like to end this presentation in acknowledging my lab back at Northwestern. First and foremost, my advisor, Dr. Luisa Irela-Rispe. She's also presented for BINA in the past. I'd also like to acknowledge my mentee, my undergrad mentee, Arya Mehta. She also contributed quite a bit to this research. Everybody else in my lab for their expertise and knowledge. And then also to acknowledge my funding bodies, the Leduc Foundation, Revamp for vascular malformations, and then also my T32, the cellular and molecular basis of disease, which I was primarily on for my first 3 years of doing research at Northwestern. Thank you.
Thank you, Katiannah. That was a wonderful talk. Wonderful, like the defense when I watched it. And it's great that we have 20 minutes of for questions. So I don't know if somebody has a pressing question. I have many questions. So
yeah, go for it.
Okay, I'll start. Feel free to raise your hands. You know.
Do you want to take down the slides and share the slides, and that way we can see everybody as well? Thank you.
Oh yeah. Okay, I'll stop sharing.
Yeah, feel free to. So for the participants, feel free to raise your hand or type if you're very shy. Type your questions in the chat, and then we will read it. So one of my first questions, some are going to be a bigger picture. What is the effect of the morphological changes that you see based on flow on their function? So do you see differences, for example, in, I don't know, leukocyte transmigration or in oxygenation of the tissues that they supply or anything like that?
So Luis and I had conversations about what the effects of altered microtubule dynamics have an effect in endothelial cells. Well, this issue about this is like, this is the first time acetylation has been studied in the context of endothelial cells. But what I can say about altered acetylation in other diseases is that this has an effect on the way they migrate, the way intracellular trafficking occurs of very important proteins in neurons and cardiomyocytes. So in trying to extrapolate, I would say in endothelial cells, it's possible that the levels of acetylation might be important in how these endothelial cells migrate in forming vessels and in the development. It could have consequences in trafficking of important vascular proteins. It's been known that certain proteins are very sensitive to flow. Like when there is no flow, it's in low levels. When there is flow, it upregulates, and it has consequences in regulating their inflammatory profiles, or essentially those kind of things, or even like identity of different vascular beds. So I can see there's a consequence in that in the context of those vascular diseases that I was talking about before, being like, oh, maybe these microtubules, while they're not functioning correctly to help these endothelial cells get to where they need to be, or even affect their function, that might have effects on their identity in the specific region that they need to be in the body. That's how I'm thinking.
Okay, that's fun.
Yeah.
That is fun. Are there any questions among the others attending? Don't be shy.
Can I ask a quick question whilst other people gather their thoughts? So this is lovely. I mean, it really is amazing work. And since nitric oxide is such a big player for endothelial cells, have you done any sort of biochemical analysis to see if there's any impact on that? Because clearly, you know, if they're experiencing greater shear stress, that basically suggests there's greater flow, and that potentially can be damaging. But if the blood vessel can dilate, to allow for that increased pressure and flow, then that's sort of a protective mechanism for it, right? So
Right.
is there some evidence that it somehow is altering their ability to produce for that protective mechanism, as it were?
I haven't actually been able to look into that personally. I was only focusing on this aspect, but like you bring up a good point, being like, what are the other functions that microtubules have in, you know, promoting or regulating these different processes that endothelial cells partake in every day? That is a very good point. That's actually a very, very, very good point. Yeah.
Sugania has a question in the chat. So it's having worked on intermediate, so she says, great talk, cool microscopy images and time-lapse videos. Having worked on intermediate filaments before, I was curious to know how intermediate filaments responded to these changes. She has to leave to a meeting, but she will listen to
Yeah,
it.
that's a really good question. So as I said before, the basically the cytoskeleton is interconnected, and what I can say is that unfortunately people are like just beginning to like start really studying intermediate filaments. They're like it's like catching on. So in a different lab at Northwestern, they found that when you completely knock down vimentin, which is an intermediate filament, they found that it affected the ability of the shape of just RPE cells. But what I found was that there are papers that have shown that when you alter microtubules, you affect the arrangement of actin. So you make a good point being like, there is probably something happening with the intermediate filaments that I'm not looking into that could also be explaining some of the phenotypes that we're seeing in this study. But the problem is like, once again, the chicken and the egg, like which one happens first? How fast does it happen? Can you really separate out the roles of all the filaments in the cytoskeleton of endothelial cell, or they probably just all work together and just a little bit of alteration just cause a cascade effect throughout the cell? Bring up a good point. So the answer is like, I believe there is an effect. I just haven't looked at it. That's the short answer of it all.
Philip is asking, how does microtubule dynamics influence the contractility of the vessel? And is the microtubule response to flow affected by aging?
Yeah, so for the first question, studies have shown that microtubules are really important for not the contraction of cells, but more for like compressive forces, like providing cell shape. So I think microtubule dynamics are probably more important in relating to cell shape related activities like migration and division, and that actin itself might be the one that's important for contractility. But maybe if the cell shape is not correct, then the where contraction should happen in the vessel could be improper. So in the case of like HHT, the issue is that the arteries and the veins are fused, and so you have this really giant mass in the center of this very like sensitive, leaky vessel. So it could be possible that the junctions between cells may not be occurring because the microtubules are not where they need to be, and that can affect contractility because then the actin can't be where they need to be as well. And then your second question, microtubule responses to flow affected by aging. Great question. Never checked that out. But what I can tell you is that someone in the lab actually showed that, at least for the nucleus, which has a lot of different cytoskeleton filaments, specifically like intermediate filaments, they found that there was different amounts of vimentin as the mice aorta ages. And so we can extrapolate saying that that's probably the same case happening for microtubules, or not the microtubule structure itself, maybe different proteins are involved in regulating microtubule stability and length could be changing over time as you're aging. Good question.
I have another question. I see that there's none yet here. So just out of curiosity, I saw a study on plant cell biology where they realized that there was an effect of gravity on whatever transport they were studying. I imagine it's the same for endothelial cells. How do you know if anybody has done something like this? Would that affect the flow work that you've been doing? I imagine compared to the actual flow, it's not. I don't know. What do you think?
I don't know. Yeah, I haven't looked in that. I haven't
So
looked
they did vertical cultures in this case and then built a microscope to be able to image vertically and crazy things like that. But
yeah, I haven't checked a look into that. I would say for these experiments, most of them were done on orbital shakers. So you just seed 6 wells, and then you have media just laying in the petri dish, and then it's just being shaken, and then we also use the abidi as well. But there are other different techniques that exist that employ gravity to drag media onto cells. And there is work showing that there are slight differences in how endothelial cells respond to those different types of flow caused by different techniques of inducing shear stress. Yeah. So I have no clue, but I do firmly believe that just the materials and the techniques and the reagents that you use also affect your experiments as well. So you could be possible. Yeah, it could be possible. Yeah.
There's another question in the chat. Could you address any developmental mechanisms where microtubules in endothelial cells are involved in the emerging curved aorta?
Unfortunately, I could not. Yes, I did not do any developmental work, and unfortunately, I haven't read any papers that also address the role of microtubules in the developing aorta. There's a lot of information on how microtubules are important in endothelial cells in forming the integrity of the endothelial barrier, but not in a developmental context. That could be something someone can do in the future. Very good question. Very important question, actually.
Well, there are more. What were imaging challenges that you faced, or what was anything that you can tell us about the imaging?
Yeah, so one of the challenges that I had in imaging was like trying to get really actual high resolution. So I had to contact a couple people about how to stain for microtubules without compromising all the other proteins, because usually people who study microtubules, they do everything in methanol, and that destroys everything else, every other protein. So I was trying to find like a balance between getting really good resolution for cell shape, but also not sacrificing the looking at those microtubules and trying to get information on like where those modifications are on the microtubules. Yeah, so we had to develop a lot of conjugated antibodies and developing our own protocol to get the best of both worlds. Yeah.
Nice.
And then for the live imaging, I had to create my own apparatus where I had to finagle the abidi chamber system into our spinning disc, and also like create our own chamber where we had enough CO2, enough media happening, and then imaging at the same time as the media is like rushing over the cells. There was so many things that we had to do. That was very difficult. Like live imaging while cells are actively being exposed to shear stress was very difficult. Yeah.
Sounds fun though.
Yeah, if anybody wants the protocol, we have it, and Lisa could definitely share it for sure for any other people interested in using a BD and live imaging.
If there's no other question, I have one that is very philosophical. So I know that that in the PhD, especially at the end when you're writing, at least I got all these ideas like I should have done that. I should have done that. That was an interesting question. So what I mean, you just defended. Was there anything that you say, this is the next step, this is what I would love to answer now?
Yeah, so that's a really good question. I think a lot of the people who have asked me questions are like, couldn't you do this and why couldn't you do that? And I was like, you're right, why did I look at it? Like there's times where I wish, why didn't I not look at the other cytoskeleton components? You know, why didn't I like use different treatments for actin or intermediate filaments and see if that affected microtubules in the context of endothelial cells? I also thought about, a little bit more about the regulation of HDAC6 and alpha TAP1, like why couldn't I do any experiments related to that? There were times where I thought about trying to actually get mouse models for those vascular diseases, and then take those aortas out and just see if there's actually an increase in acetylation or any changes in microtubules in those diseases. There were so many things. And then also, at least in my case, you think you read all the papers in the field, and then you are writing your thesis, and you see how many papers you missed while you were writing that could have answered a lot of questions for you and probably helped you along the way much faster. That happened to me as well. Yeah.
It happened. I mean, it's impossible to cover everything, right? You have a limited amount of time. So, but yeah, there's always lingering questions. I was wondering what that was.
Yeah.
Great. We have 7 more minutes. So if there's more questions, feel free to ask. Otherwise,
thank you, everybody.